Namaste, respected dignitaries, Principal Ma'am Dr. Anuradha, our eminent guest speaker, Dr. D.P. Saini, dear colleagues, and my beloved students. Happy morning to one and all. This is Dr. Netra Subramania, your MC for the day. Welcome, Sri Narayana Sir, Managing Trustee, Padma Shri Group of Institutions. Sri Satish Sir, Chairman, Padma Shri Group of Institutions. Our beloved Principal Ma'am, Dr. Anuradha, Padma Shri Institute of Management and Sciences. Today's eminent guest speaker, Dr. D.P. Saini, Founder and CTO, Protein Design. And all the participants to an industry expert talk on protein design organized by Padma Shri Institute of Management and Sciences. Vakratunda Mahakaya Suryakoti Samaprabha Nirvignam Kurume Deva Sarvakaryeshu Sarvara. With these prayers, let us begin the session. Now, I request our Principal Ma'am to give a formal welcome to our guest. As we all know, protein is an important building block of life. So every cell in the human body contains proteins. The basic structure of protein is a chain of amino acids. Protein help our body repair cells and make new ones. Protein also help for growth and development. The scope 1 field, proteomics, also help us to analyze proteins, structures, and its production. More insights about the topic will be given by our eminent guest speaker today. Now, I request our Principal Ma'am to address the gathering on this occasion. Over to you.
Good morning, everyone. Energy is little low. Good morning. In this particular program, I don't want to stand between you and Dr. D.P. Saini ma'am. The purpose of starting this kind of initiatives, all these days we are working online, and we don't know the importance of physical interaction. I want you to know how companies are working and where you can go and do your jobs or internships or placements, whatever so it is. But it is always very exciting and electrifying to listen an industrialist who is really good able to turn the knowledge into money or knowledge into applied aspects and doing the service to the humankind. They are having excellent laboratory, and anyway, you will be listening to her. Definitely, I am sure that you will have wonderful session today, and this session is only for 1 hour. After that, you can interact with her, and finally, we will take it forward. I will tell you how we are going to take it forward after her talk. Anyway, have a wonderful session. Good morning once again, and have a nice day.
Thank you very much, ma'am, for that introductory part of the occasion. Now, it's time to listen to our guest speaker, Dr. D.P. Saini, Founder and CTO, Protein Design Private Limited. Ma'am has PhD from AIMS. She was a postdoctoral fellow from Department Pharmacology and Toxicology, University of Arkansas for Medical Sciences, USA. She was also a postdoctoral fellow from Department of Surgery and Pathology and Immunology, Washington University, USA. She has also worked as principal scientist, Genotypic Tech Private Limited. Ma'am has been awarded with many awards, like she is being awarded as Young Investigator Award at the annual meeting of American Transplantation Congress. Best Oral Presentation at annual meeting of the Indian Immunology Society. Senior Research Fellowship awarded by CSIR. Junior Research Fellowship awarded by CSIR. And even ma'am is a rank holder in her UG and PG. Ma'am is also published many of her articles in the reputed journals. She has published her books, and also she has participated in many conferences and symposiums. So, coming to her professional experience, ma'am is having the research experience which includes the screening of phage display, SAFP library for isolating clones, selectively binding to antigens over expression of the clones, and purification for kinetic studies and cross-reactivity analysis using various immunological methods and animal models for in vivo analysis. And basically, she is very much experienced in the protein field. With immense pressure, on behalf of Padma Shri Institute of Management and Sciences, I welcome you, ma'am, to address the gathering.
Good morning. I'm very happy to be standing here in front of you today. Thank you so much to ma'am and Hindu ma'am for this opportunity. Can you hear me at the back? Okay. I would have finished the talk without you guys ready. Good morning. I would like to thank Principal Ma'am and Dr. Hindu for giving me this opportunity to interact with all of you today. I'm really happy to be here. I hope you guys will be happy by the end of the hour. It's good to be offline and talk to you rather than talk to a screen. So rather than a boring technical lecture, I'd like to tell you a few stories. Story of our lab, story of protein design, and how we ended up doing what we are doing. So, and how we can do this together in the lab. See, learning theory and getting degrees is one thing, but to actually be at a lab and do the experiments towards making products and things like that is another thing, right? So how you guys can come to our lab, I can be here, we can be here and do things at lab here. So I'll try to make it not a very technical talk. Please feel free to ask questions. We don't have to wait till the end of the session for questions. I don't have so many slides. Okay, more pictures. So yeah, our lab is called Protein Design Private Limited, and it's incubated by the Society of Innovation and Design at IISc Bangalore. So proteins. Of course, apart from amino acids, what is the other things that come to mind when we think proteins? Can I have a few answers? Louder, please.
Chicken, egg, it's in.
Well, I'm thinking more like enzymes. Yeah, like hormones. Yeah, antibodies. Right. The main things that come to mind for a food person and for a nutritionist, it might be a little different. For a person who's involved in healthcare, it might be a little different.
Protein of COVID-19.
Yes, the spike protein of COVID-19 used for detection in the antigen test, and then of course it has been used as the antigen for making the vaccine. Yeah, so yeah, vaccines. I mean, everybody is aware of all these things. So these are some of the products coming out from pharma companies and the Serum Institute and Bharat Biotech has made all these vaccines. So everybody is kind of aware of how important proteins are. Yeah, hormones for that matter, reproductive hormones, growth hormone, those for therapy, immunotherapy towards cancer with antibodies, diagnostics with antibodies, the ELISA testing. Everywhere you see there is proteins and enzymes, not to forget. Whether it's now enzymes are there in shampoo, there in detergent, there in anything that you want to do, they're trying to make it more organic, and proteins and enzymes will play a big part. So this whole list that you are not seeing all because I have slides for that. Oh, all this while I thought we had the proteins on the screen. Anyway, we said all the things that are on the screen. So, yeah, on the whole, protein design works on proteins from natural sources, recombinant proteins, hormones, biosimilars, antibodies, etc., etc., etc. I've already mentioned. Does everybody know the story of insulin? Right? Insulin, the hormone used by diabetics all over the world. Yeah. So initially when they started, where did they get the hormone from? Exactly. They go to the slaughterhouses, get big pancreas, make a paste of it, try isolate from there, and then it was put into the formulations and so on. And what happened? It helped a lot of people, except people had immunological responses against the big protein. The batch to batch variation was there depending upon how long the pigs had been there, how it was processed, and all those things. And then came in recombinant DNA technology, right? So they said, oh, why big insulin? Let's make human insulin, right? So the first product of modern biotechnology, and a good example in all the textbooks, is insulin. Everybody knows it's made of three peptides, one of them is cleaved, and then the whole insulin molecule works. And so just like
just check, go front and back. Go next slide and check. Next slide and check.
So reproductive hormones is another one of his strengths. My PhD was to do with recombinant antibodies, SCFEs that you heard in the introduction about. So antibodies are my favorite molecules. Okay, that's how. But then I worked in the field of biochemistry, toxicology, immunology, applying all these proteins in clinical studies, also in diagnostics, and so on and so forth. So there is a whole lot of talk about genomics, transcriptomics thing, but I feel proteins are the real soldiers. They are the actual doers in the cells. And proteomics, metabolomics would give you a more realistic scenario of what is going on in the cells or tissues. So that is the kind of field that we are working in. So yeah, the lab is at MSR Nagar, Mathakere. It's right next to the ISC campus and close to the MS Ramaya Hospital. So I'm sure you all will visit and see our lab. It's not like I mean I know it's an industrial lab, but it's not huge. But I mean we are pretty well equipped and well set up for doing recombinant proteins. So technically we have kind of 3 labs. One is our molecular biology and microbiology lab, where we do all the stuff with cloning, enzyme assays, colorimetric assays, and things like that. Anything to do with DNA, RNA, and proteins. Also, we handle the microbiological, the bacterial system for protein expression. So here, I put a picture of DNA, RNA, PCR products, all those. There is pictures of plates showing bacterial colonies. There is a RT-PCR kind of output, QRT-PCR. Everybody knows RT-PCR and QRT-PCR now, right? Because COVID diagnostics is done by QRT-PCR, fluorescent-based assays. So those we do at the lab, not diagnostics, not COVID, but other gene expression studies we do. So a picture of some of the instruments and people in the lab, you will come and see and also meet. So yeah, I mean, like I said, I put in point-wise all the things that I listed out, doing DNA work, RNA work, PCR, amplification, the restriction, cloning, transformation, all those things we do in the molecular biology and the microbiology lab. Toxicity testing, we do food assays, antioxidants, polyphenolics, adulterants, contaminants, so those kind of assays also we will run in the molecular biology lab. Then this is an overview of our yeast lab. So we work with Pichia systems, Saccharomyces. These are some of the common yeasts that are used for fermentation process, and these are good hosts for expressing recombinant proteins. Vectors are available, and you can do a whole lot of cloning and expression work. So again, this whole I'm talking about my yeast lab and the cell culture lab is showing up. So yeah. Yeast colonies on the plates, they're different from bacterial colonies. So once you get the lab, you will see how things are a little different handling the bacteria as well as how things work in yeast. The yeast hood, we have the centrifuge and the shaker in which the yeast grows. Yeast grows slower than bacteria, okay, but it produces a whole lot of protein just like bacteria. Why would we want to do some proteins in yeast? What do you think will be the difference between making a recombinant protein in bacteria versus yeast? What's the one word answer? What is the difference? A little louder. Absolutely. Yeast is eukaryotic. So post-translationally modified proteins won't be modified in bacteria. So anything that depends on its activity or for its structure on post-translational modifications will not come out well from bacteria. Makes sense, right? Yeah. The other thing is do it in mammalian cells, insect cells. These are good systems for expressing mammalian proteins, right? What is the problem? What is the problem with expressing proteins in mammalian system? It's a good idea, right? You will get more human-like or mammalian-like proteins. Good post-translation modification, good folding, good activity, everything, right? The scaling up, the growing of the cells, they grow so slowly, and you need really sterile aseptic environment for them to grow. One bacteria in there, it will all become dahi, isn't it? Correct? So culturing mammalian cells is tedious, expensive, slower, and then to get protein purified from there is also a little difficult because the culture involves growth with serum in there. So there's a whole lot of serum proteins and background proteins that you have over there, right? So yeah, but then if you want to do an immunotherapeutic antibody, of course you can't make it in bacteria. It has to be made in cell. So in the mammalian cell, which is why each of these injections cost like lakhs, right? The cancer patients have to have these immunotherapeutic injections, which cost so much. And of course, the big pharma, the R&D companies have spent so much money making them, and they have to recover all that money from the patients. So it is so expensive with their patents, given to these companies. But over time, we can have, indigenization, or we could make it at a scale larger than what they make, or at a cheaper cost. Wouldn't it be wonderful? Right. So those are some of the goals of any R&D company, especially geared towards pharma or healthcare. Okay. So, same thing with industrial enzymes. Anything that can be done slightly cheaper or at a larger scale or a better purification or a better yield is good modification. So all that involves protein design and protein engineering. Okay, so the cell culture lab we have a carbon dioxide incubator, a cell counter, a microscope, of course the food and everything, and media and all we keep over there or whatever. All the setup for cell culture lab, which will be set up over here at the at your institute also. Okay, so yeah. So what are the three labs we have? We have the bacterial microbiological lab, the yeast lab, and the mammalian cell culture lab, which means we have 3 systems to make recombinant proteins. Right? So the design of the experiments is such that we choose which one depending upon what protein we are trying to make. Okay, and then comes the downstream process. Once you have the recombinant protein made in the host, the whole thing about purifying it with chromatography. Okay, and estimating quantity, quality with STSA. Everybody is aware of those techniques, right? You and of course, now that you will be back to lab, you'll be doing more practicals and hands-on these techniques. Okay, if you want more practice, you can come over to our lab and also practice more. Do and then we'll talk about how internship or projects can be run at the lab here at your lab as well as others. Ma'am will talk about that later. So yeah, and then ELISA, Western blotting for characterizing the protein, mass spec-based characterization, all of those we run. We have an ACTA system here, the machine there. It's an automated protein purifier, which means we can set up the chromatographic column, put in the program for it, and the fraction collector collects the flow through, washes, elutions, etc. automatically, and then you can analyze all that for how the profile of purification looks. So all this under the downstream process. Okay, so now 5 minutes for telling you about what is special about protein design. So like I said, we specialize in protein expression, bioassay development, and validation. This is our strength, and customization for the requirement of the project. Antibody development and humanization of antibodies, which means you get the variable parts, you put them on a human framework. Humanization of antibodies. That is something that we can do by recombinant technology. Then receptor ligand interactions, expressing receptors, seeing the effect on cells, cell-based assays, and stabilized generations for doing those kind of assays. But the 3 patented technologies that we have is the Synvac system. We call it Synvac. We have to probably name it because it sounds like another thing. Is a strategy where we can vaccinate with a single dose without needing any boosters with very low antigen. So this is a patented technology we have, and we are working on making this a product for now with the help of DBT and a small brand that we have at the lab. Then the second is glyco-engineered yeast for protein. We did say yeast is good. The wild type yeast does post-translation modifications, but the glycosylations are different from eukaryotic. They're not exactly the same. So what we have done at our lab is we made a yeast strain with a couple of enzymes added in there so that it can cleave off the glycosylation, the extra whatever the yeast does, and add the final residues just like the mammalian cells. Okay, so we have a patent for that glyco-engineered yeast strain, which makes human-like proteins. The best part is it will overexpress like bacteria, and it will do post-translational modifications like mammalian cells. So it's like the best of both the systems. And then, of course, our experience with protein engineering and protein design to customize any kind of protein product that can be made. So that's about our lab. I mean, we have a few human growth hormones, all these products, insulin and derivatives, PDH, glucagon, some of these we've done already for clients and for our portfolio. But then, anything that recombinant proteins are required, we can customize and make in all the three systems: bacteria, yeast, and mammalian cells. Is everybody still awake? Yes. Okay, they're all good. So the services are basically customizable. An overview of what I said so far: cloning and this thing in the system that is required. Depending, see, proteins are small, big, homodimeric, single unit, multimeric, heterodimeric. Basic proteins, acidic proteins, neutral proteins, globular, fibrous. Everybody knows, right? So many different functions, so many different structures. So depending upon what protein you have, which system you will express it in, what vector you will use, what tag you will use, what selection marker, how will you do the downstream processing, all these stories are protein design. So with all this in mind, we named our company Protein Design, and that's what we basically did. So then making a strategy is one. The second step is expression optimization in the system that you require, and then purification. Protein purification is science when applied to one protein. For every protein, it's different, so it becomes art. The more proteins you see, you will realize that every protein, just because you purified one protein, doesn't mean the same protocols will apply to the second one. So this is what is involved in designing your experiments and optimizing them, and then of course carrying out and producing good protein, pure protein, active protein. Making a whole lot of protein. Overexpression is easy. Reduce it, push the cells, host cells, proteins. Showing the enzyme activity or
third sem
Right. So the whole point is structuring experiments in such a way or designing it in such a way that you get good protein, enough of it, and okay, so that's the thing. Yeah, I'm going to talk about protein engineering and life sciences. Eventually, you all know whether it is agriculture, biofuels, polymers, flavors and fragrances, especially applied to food. Everywhere there are proteins involved, and they require more and more refining. There is enough helping and say, I mean, please don't read the whole slide. I don't know why I haven't put that much part on it. The saving and extending life applies to all the proteins used in healthcare, for diagnostics and for therapy. Feed the world. Food is the most important. Now, single cell proteins, the enzymes, all sorts of supplements. We need more and more and more with more and more people in the world. Then, environment. Bacteria, which they chew up plastic now. Oil spills, if bacteria can take care of them with their enzymes. Wonderful. If we can do any of those things using recombinant technology or enzymes made there, that would be so helpful. So yeah, some of the examples of applications of engineered proteins: amylases, catalases, cellulases, invertase, lactase, lipases, invertase, and zymase. Where is that applied? The food guys. Alcohol production, guys, beer industry, wine industry, isn't it? And then now ethanol is coming up as a big biofuel. So there are so many applications of these enzymes. There is so much competition to make the best strain of fermenting, the thing yeast, which can make maximum amount of alcohol because there's so much demand. And now with sanitizer use and things, I mean everybody knows ethanol, isopropanol are like the most in demand molecules. Okay, food industry. So proteases, amylases, lipases. So all of these enzymes that are important can be made recombinant in a recombinant way, also engineered to do better in terms of their stability, in terms of their activity at room temperature. See, we put enzyme, they say this enzyme enriched detergent. It'll take care of your greasy stains, it'll take care of your colored stains and things like that. But with so much detergent means alkaline pH, right? All proteins enzymes will die. So you have to make enzymes which will work at pH 9, 10. Correct? So those are the things that we can do with designing proteins or engineering proteins. Again, proteases are used in several applications in food industry, especially with low allergenic infant formulas. You don't want babies to get allergic responses. Then alkaline pH, low temperature. The proteins should be stable in those kind of environments. Amylases again, both for food and detergent industries, for flour, for bread making, cake making, all that in the baking. Again, same thing. Lipases, they have to be more stable to be used as emulsifiers, especially when you're making cheese and so on and so forth. Okay, all this is available on Google, on Wikipedia, in your textbooks. So, I don't need to go into details of all this. And of course, environmental applications of proteins, dyes to help with pesticides, biofuel, biorefining. There are, of course, a whole lot of medical applications of recombinant proteins and engineered proteins. Okay, biopolymers. And in nanobiotechnology, this is of course one of the hot fields, and I'm sure there are students who are interested there and have read up about all that. So again, let me not go into details. Please look up the examples. Ask Google Auntie; she will tell you all about it. Okay, redox enzymes. Other applications of enzymes in viral now infectious diseases and things like that. So, and more protein-based drugs that can be made with a improved half-life and less toxicity, and all that can be used for making multi-drug resistant kind of thing, to counteract the multi-drug resistant pathogens. Okay, so yeah, some of the ones that are important for human use, for healthcare, and they have made money. Insulin, of course, the biggest one. And if you do get a chance, please visit Biofund. There now they started making insulin in yeast systems. Initially, it used to be in bacterial systems. Their fermenter is like 400,000 liters. It's like those water tanks in a city. You have to go up the stairs to even look at the fermenter. It's like huge because they make so much insulin for everybody, like in India as well as for exporting it. So yeah, that's the scale. It is a $1 billion industry, and Madam Shaw claims that she will make it available at what, 7 rupees per unit kind of cost. So which means you have to make so much of the insulin. Erythropoietin factors, the blood clotting factors, all of these are so important for saving lives. So yeah, I've been kind of talking about protein engineering. How do we do it? We know proteins are made from transcription and translation of DNA. So if we change the bases in the DNA, the proteins will differ. Correct? So this, what is this process called? Mutagenesis. Right? How can we do it? How do we do it? Any ideas on how to do mutagenesis?
Chemical treatment.
Physical ways, chemical ways, radiation. These are general. Everybody knows mutagenesis causes cancers also, no? UV radiation causes radiations, chemicals, certain carcinogens, they'll all trigger. So you can do random or a whole system kind of mutagenesis. You put your construct in a mutator strain. It will give you many clones. Then you check which one makes more active protein or gives better yield of proteins, and you can screen like that. Or you figure out which amino acid in the protein is very important for activity or for stability. You change that one amino acid and then see how the protein changes. Right? So specific site-directed mutagenesis we can do random mutagenesis, we can do many changes and see how the structure of the proteins are changed. This is all involved in protein engineering. So of course the important is the host and the vector. The vector will carry your gene of interest to make your protein of interest, and the host will transcribe and translate it. Right? So the vector is important, the host is important. There are so many vectors available in bacterial systems, so many in the yeast system. Mammalian vectors are different. Their antibiotic markers are different. Their promoters are different. Right? The kind of cloning you will do will also be different. The way you put it in the host is also different. What is that process called? Putting your recombinant construct into the host. What is that called? It is the overall insertion, but we call it transforming the host. Right? In mammalian system, we call it transfecting the host. How do we do it? Right from 12th grade biotechnology chapter we are studying. How do we transform the host? Come on, one answer at least. How do you put your recombinant construct into the host to make a recombinant protein?
Plasmid.
Plasmids, transformation by making competent cells. So chemical transformation. Electroporation by zapping the cells, take up the plasmid. Injection for IVF and things like that. Right? And in mammalian cells, we do transfection using liposomes. So the liposomes can carry your recombinant DNA and take it into the mammalian cells. Right? So vectors are important, host is important. And yeah, this one slide is actually the overview of everything that I have said. The transgenic animals to make protein, plants as expression hosts, mammalian cells, insect cells, yeast, bacteria, all the host systems. So they can all be compared in terms of speed of producing proteins, cost, the glycosylation and post-translational modification, the folding, and then of course the regulation or the rules governing this. So in terms of speed, bacteria is fastest. You put one bacteria in the medium in the night, morning, full log saturation phase done, lag phase, log phase, back to the saturation, right? Yeast take about 3 times more time. But again, in 2 days, you will get a good culture. Mammalian cells. 1 week it will take for them to grow to become like 4 times. Okay, so speed in bacteria is fastest. Cost also, bacteria is lowest. What medium do they grow in? LP, urea broth, everybody's heard of it, right? Yes? So it is very easy to make 2-3 components, autoclave, steam, sterilize, and it's ready for this thing. Yeast also not so expensive, but the carbon source is usually a little more expensive, and you need some amino acids, etc., as a supplement for yeast medium. There is one more big difference. Bacteria need salt, whereas yeasts need sugars to grow. Okay, so medium is slightly different, cost is little bit different. And when it comes to mammalian cells, the cell culture medium, buying that only, you have to think like 10 times to do mammalian expression. It's like 10 times more expensive than your bacterial medium. Plus, you will need it for a longer time because the cells grow slower. So cost in terms of doing it in mammalian cells and animals, transgenic animals making proteins there is exorbitant. But then again, depending upon what you want to make, if you want to make immunotherapeutic antibodies, you have to make it in mammalian cells or biosimilars. So then glycosylation is worse than right? They don't do any post-translational modifications. It's slightly better in yeast, but then if you want perfect, it has to be in mammalian cells or transgenic animals. Holding of proteins obviously will be best in mammalian cells and worst in bacteria. So if you want to make a whole lot of protein, which you want to use as an antigen for making, for raising antibodies or for a diagnostic purpose, for a kit like that, bacterial expression is easy, quick, cheaper, and it will serve its purpose. But then again, if you are making an enzyme, you are making a hormone which has to do an activity. In bacteria, it won't really give you a good protein. And then, of course, the easiest, the RCGM and the committees which are involved in regulating recombinant DNA work, because it can be used pretty maliciously, is easiest for bacteria. You can get permissions easily, and it is more difficult with animals and plant. And actually, now that some proteins have started to be made in yeast, things are opening up, and yeast is being accepted as a good host for proteins. And eventually, I really strongly think that a lot of proteins will be made in the yeast systems. So this is kind of how we plan how to make our recombinant proteins. And we've already talked about this. Upstream processing is all the process you're doing till cloning and getting the host system ready for making the proteins. Once they have grown, the downstream processing involves getting your host. Okay, proteins can be intracellular and they can be secreted out. Correct? So if your host is making an intracellular protein, you will do the purification from the cell mass. Whereas if it's a secreted protein, then cell mass is best. You'll use the spent medium and purify the protein from there. Right? So all those steps are downstream process, where you get your source of the recombinant protein, purify using chromatographic techniques, etc., test for quality, test for purity, and all that, and then the drug product or the protein product is made. Okay, so yeah, basically cell disruption, purification, and making sure that its quality is say 95% pure or whatever is the demand of the industry. It is done, basically this needs the most optimization. So now chemical engineers and biotechnologists, and there are specialists who are or experts who look into optimization of fermentation or bioproduction or downstream processing. It's a whole R&D department in itself in every company making protein products. Okay, how are they different from small molecule drugs? How are protein products different from small molecule drugs? Can you name a few small molecule drugs? Plenty memes going around since last year about a small molecule drug. Can you name it? This COVID wave, there were so many jokes, memes about a small molecule drug. For any problem, take Dolo. Right? Aspirin. These are all small molecule drugs. They're chemicals. So they are very different from proteins, which are biomolecules. Right? So the industry which is geared towards making small molecule drugs, it involves not so much optimization or not so much. I'm not saying those are not important, but the kind of R&D or optimization or design and engineering that goes into making protein products is very different from small molecules, because their purity, stability, etc. are easily assessed also, assayed also. Whereas with proteins, every batch you might get variations and has to follow optimal QC procedures at every step. Okay. Then, of course, they are more temperature, pH, buffer. All these are very important for peptides and proteins. So you have to, watch about that also. Proteins have different size, charge, holding, glycosylation, activity, temperature, and pH sensitivity. I said all that. Yeah, fermentation process. Or how do fermenters and bioreactors work? You scale up what you have optimized at small scale in the lab to a large scale. It doesn't always happen like this. There are so many molecules which work so nicely, get so nicely purified at small scale, and then you make a 50-liter culture or a 200-liter culture, you don't get the same kind of protein expression, the same kind of, purification. You'd expect that if you scale up everything, scale up, it will work accordingly. It doesn't. So then, there is a whole optimization involved in scaling up to a fermenter process where you can say do it in a 20,000 is the biggest one put over here. If you have to do a fermentation there, then how much aeration is going to be, at what speed you're going to mix it, how often you're going to feed the cells, media change, how long the induction will be. Everything has to be optimized. So proteins are very interesting, tricky to work with. Okay, and it has to be sterile, because you don't want anything apart from your host to grow. You don't want any bacteria or yeast coming from the environment, from the water, from everywhere, and spoiling your culture. So aseptic environment is very important. Now you have use and throw fermenters. Fermenters are coming as disposables now. At up to even 1,000-liter kind of volumes. So if you get a chance to visit an industry where they do fermenters, Biozine is one of the companies. Industrial visit to there, would be awesome. Go to a distillery, see how they do the fermentation. Go to Biocon, see how they make the fermented protein. So if you get a chance, visit these industries and see how these products are made at large scale. Then, of course, purification. In lab, we do an experiment. We'll set up one column. We'll put our mix. We'll separate out and things like that. You want to make billions of thousands of units of insulin. Means small columns won't work. The columns itself are the size of a room. So everything has to be upscaled to that much. So this is how a purification process also looks. First, to make sure your host cell mass or the medium is ready, whether you'll run it through ion exchange or you'll run it through apoptotic chromatography or hydrophobic chromatography. Depending upon that, the systems will be set up. Step-by-step purification will happen, QC at every step, and then finally your product will be formulated and aliquoted and dispensed. So again, I already went over this. This is what happens when you don't follow the slides and try to tell a story. Mutagenesis, I already said that you can do a random one and then pick up your interesting clone, or you can actually have a strategy bioinformatically or in silico and then apply it to lab to do specific site-directed mutagenesis and make the changes to see if your proteins become higher yields, more stable temperature. It will be so nice to have an insulin which can be transported at room temperature, isn't it? It won't spoil, and people can take it while traveling. Otherwise, you have to carry an ice pack to carry your insulin when you're traveling. And it's so inconvenient. And anyways, carrying the needles and all is not convenient at all. And especially when you have to take it like 3 times a day. Right? They're trying to make an analog of insulin. It's called glargine. What is special about glargine? It's a slow release. So instead of taking like 3 times a day before every meal, you can take it once a day. Now, probably they'll come up with something which you need to take only once a week. It will be so convenient, isn't it? Otherwise, injecting here, every meal, painful. So basically, engineering of proteins or biomutagenesis or designing them involves changing or optimizing in such a way that the properties of the protein become better. PCR-based cyclic nutrients, again, no need to go into technicalities. You studied in theory, and if somebody wants to do at the lab, we can do in the molecular biology lab here or at our lab. It's very, very possible. Random mutagenesis also by chemical treatment or putting in a strain where it makes mutations can be done. Okay, so now that we know what protein engineers do, what protein chemists do, downstream processing people, quality control people. So there are a whole lot of career opportunities for biotech graduates or food technology graduates in the field of say pharma, diagnostic, medical lab, agricultural industries, in beverage and food production, research laboratories, R&D labs, hematology, bioremediation kind of setup, immunology, forensic sciences. It's really important. Again, if we know DNA recombinant technology or protein-based diagnostic procedures, whether it is ELISA or RIA or whatever is applied, then in biological processes like crop development, pest control, wherever applications of biotechnology are, in veterinary services, animal husbandry, animal production, and then of course anywhere in a molecular biology or a protein engineering lab in any kind of an R&D setup. So these are all the kind of jobs or career opportunities available to you all budding researchers and scientists. I see the potential there. So which is why I'm happy to be talking about what all can be done. The careers can be wet lab based or dry lab based. Dry lab bioinformatics basically, or where you do all sort of in silico analysis and plan or design things, and then of course it has to be tried in the wet lab. So that is bioinformatics is a hot field now, and you can increase. See, at college you learn a whole lot, you do a whole lot of practicals, but always to enhance or complement those, there are internships available in industries in academic settings, and there is a BCIL program, the industry program from DBT, Department of Biotechnology India, and then Karnataka government has the biotech skill enhancing program, which is a 1-year kind of program. 6 months you do training at the college, which is involved with the BICEP program, and then 6 months you spend in the industry learning how things work, so that you're job ready, and you get a better opportunity than just a fresh grad, than just having your degree. So it always helps to go see or spend time in labs and see how the industry works, how academic labs work, so that you can get practical exposure. Okay, and yeah, that's my last slide with my email, phone number there. Please feel free to get in touch through your college directly. You can send emails, you can WhatsApp, you can call, come to the lab, talk to me here, talk to ma'am. Okay, so thank you for your time and patience, and if you have any questions, I'd be happy to answer.
For an excellent overview of the entire world of proteins. This is a very, very, very important segment. I would like to tell you all, my dear students, connecting the dots is very, very important. Whenever we see that in the lab, when you are doing something, you don't connect the things what you have learned in biochemistry lab and what you are going to learn in molecular biology lab and what you are going to do it in plant biotechnology lab. Please don't have that kind of mental makeup. Connecting the dots is very, very, very important. When we say that you have to learn this, don't learn it like a technique. You may become a general manager in an R&D. When you become a general manager in an R&D, if you have a problem, at least you should know how to resolve it, to which lab you have to send for an analysis, and how to design your entire product line. Otherwise, you will be lost. Don't study only for the sake of your examinations. I know they're all important enough. If you study for 1 week, you can get above 75% marks if it is only for your examination sake. Don't do that. When she's talking, you might be having so many unknown words. How many of you know about biosimilars? Have you heard about it earlier? Please be truthful. If you are not, there is a platform to discuss with people like Dr. Deepti. I want you to interact, open up, try to learn. Even in this age, it feels like learning. You know that how many protein products are there in the market? Forget about therapeutics, forget about drugs, forget about just get into, if you go to people who are suffering with cancer after chemotherapy, just doctor prescribes, you have to take whey protein as a supplement because you need lot of protein supplementation. Have you ever seen the price of it? Have you ever seen one small box with the smallest pouch inside will be costing you around 2,500 rupees. This was my dream, my dear students, to give cheapest protein to chemotherapeutic patients, which is all there in your hands. Soya protein we import. Do you know that we import soya protein with this kind of energy, with this kind of manpower, with this kind of industry setup? When we say make in India, you feel like making watches, making laptops. But first thing is food, right? First thing is food. How much protein products are getting wasted in the agriculture field? 90% of our food is getting wasted in the agriculture field without getting processed. And many of them are allergic to some proteins. Many of them are allergic to some proteins. You have any lactase imbibed ice creams or coa? Indians may not need it, but export it. That's where you get your money, you get your job. Have you thought about it? Think in those lines. Connect the dots. Please connect the dots. If you are not good in connecting the dots, okay, some talk is being organized. We'll have fun for 1 hour. After that, we will just have some biscuits and tea and get away from this place. Will not take you anywhere. I don't say that all of you are like that, but leave your inhibitions. From past 3-4 days, we are continuously organizing something, and not even a single question is coming out from students. Don't hesitate. There is no stupid question. There may be a stupid answer, but there is no stupid question. Please try to ask, introspect, and try to ask questions, interact with people like Dr. Deepti. Once again, I thank you for your electrifying speech. And I hand over, before I hand over mic to Netra, I would love to tell you that she opened up. She is willing to share her laboratory facility. If you want any kind of a training, you can talk to your class teachers, your HODs, and your vice principal, and come back to us so that you can go not only for an industrial visit, just 1-day industrial visit will give you an idea. When she asked a question about what is the important protein, such a small, tiny spike protein could be able to jolt the entire world. Yes or no? That spike protein is the one which is causing serious infections. It could be able to jolt the entire world. Even a lay person is talking about that. Everybody is talking about that. Illiterates are talking about that. 2 years we could be able to take that small, tiny one, which you could not able to see with your eyes, could be able to make a halt. Just a break to the entire world, right? It's such an important thing. I have people who are asking us if you can give me some vegan protein as a food supplement, if you can purify some therapeutic protein from turmeric. You know that turmeric is having turmeric. which is a therapeutic protein which goes into the gutters. Before coming, the turmeric what you get into the market is being boiled. It is water soluble. Turmeric goes into gutters in the field itself. We don't even know. Such a value addition to the thing, you know. Please try to think. Don't think only from 9:30 to 4:30. Think beyond that. Think beyond your classroom. Think beyond your subject. Think beyond your practical class. And so that you will not come to us, show us a job, show us an internship. You will find it on your own. I expect all of you to come to us. Ma'am, I have 4 offers. Which one I have to choose? Try to guide us. Which is the best offer? To which company I have to go? I want all my students to be in those lines. With this small note, over to Netra. We have one more small event, and after that, we are winding up this session. Thank you, one and all.
Thank you very much, Principal Ma'am, for adding your points to an effective and informative presentation by Dr. Deepthi. Actually, Dr. Deepthi gave like more insights about how to design a protein, and with an excellent example, that is insulin, and also ma'am highlighted about how like the molecular biology lab, the microbiology lab, yeast lab, cell culture facility works in protein design. Ma'am also highlighted about different technologies that are used in protein design, and ma'am about what the company offers and what are the services offered in the company. And she also spoke about the protein engineering, which is literally an upcoming field where there are different career opportunities. And also she explained about its significance and different applicative parts of protein engineering, that is at food industry, environment, and biopolymers, and even in the nanotechnology. Thank you very much, ma'am, for such an effective presentation, giving such an insightful presentation. On behalf of Padma Shri Institute of Management and Sciences, I am happy to inform you that our institution is tying up with protein design for the research activities, and we are signing the MOU. So further information would be told by our principal manager.
Already signed an MOU. We are going to exchange the MOUs. This is for internships, training, and placements, and also for having collaborative courses or programs, value-added programs between Padma Shri Institute of Management and Sciences and Protein Design with Dr. Deepthi Saini.
Thank you so much, ma'am. Now I request our principal ma'am to do the honors to Dr. Deepthi. We have come to the conclusive part. On behalf of Padma Shri Institute of Management and Sciences, I would thank Shri Nayanappa Sir, Managing Trustee, PGI, that is Padma Shri Group of Institutions, Shri Sati Sir, Chairman, Padma Shri Group of Institutions, our beloved Principal Ma'am, Dr. Anuradha M, Padma Shri Institute of Management and Sciences, today's eminent guest speaker, Dr. Deepthi Saini, founder and CTO Protein Design Private Limited, and the organizing committee and all the participants. I would like to thank one and all for making this occasion successful. Have a great day. Thank you. So there would be a feedback link being shared by your class teachers. Please, I request all of you to give your valuable feedback. Thank you, one and all.